Hi guys, this is Mrs. Mendy, and today we're going to talk about Punnett Squares. And Punnett Squares are a technique, a way to predict what babies will look like. And they could be baby anything. They could be baby people, baby dogs, baby fungus, baby plants, is what it was developed for. Okay, let's find out. Okay, so it's a kind of mathy looking thing. And it predicts how likely two creatures, anybody so long as they have two genes for or two alleles for each gene, how likely they are to have different kinds of offspring. And it'll kind of look like this. You don't have to worry about that yet, but it's coming. So, just a little review because there were some things you will need to know. All these vocabulary words that you'll need to know for doing this. That's why we did them. Okay, so these eyes on this cute baby are brown. Is that their genotype or their phenotype? Okay, it's phenotype. Phenotype, remember, is pH, it's like the physical body. Ph, physical. Ph, phenotype. And that's phenotype is what it looks like. So brown eyes is the phenotype. So if I ever ask you, what is the phenotype of something, you will know. Okay, another quick review just so you make sure you know these because you will need to know this kind of stuff to do the Punnett squares. Okay. Gamete, we haven't really talked about. That may come up, but that's a sex cell. So it's a sperm or egg. That's how you get the genes to the baby. Okay. Allele, remember, is a flavor or a type of a gene. The genotype are the types of genes, the types of alleles, really, a person has. So, like big T, big T, big T, little t, little t, little t, like that. Phenotype are the traits that show. So that's what it looks like. Is it tall? Is it short? Is it purple? Is it white? Okay, dominant. Dominant is the stronger allele, sort of stronger. And that's shown by a capital letter like big T. Recessive is the weaker allele. That's the one that is overpowered and doesn't show in the heterozygous like this. So that's the lowercase letter is the recessive one. Homozygous, remember, means two of the same alleles. Heterozygous means two different alleles. Um, what we'll be doing is called a monohybrid cross. I won't test you on that, but just so that you know, that's what it's called. And that's when you're crossing one pair of contrasting traits. You can do it with multiple ones, but we will not do it this year. You may do it later along in your biology career. Okay, an offspring, remember, means babies, basically, even if it's from a fungus and you don't think of it as a baby, but it's a baby. Okay, double checking that you get this. These blue eyes are little b, little b. What is the genotype? What is the phenotype? Okay, so genotype, that's little b, little b. The genotype is the types of genes. Okay, little b, little b. The phenotype is blue eyes. Okay, phenotype's what it looks like. Genotype is what the genes are. Okay, a Punnett square is a way of figuring out what offspring two parents can have. So let's think of this as an example. If we have a blue-eyed person, so that's their phenotype is blue-eyed, and their genotype is little b, and you cross them with a person whose phenotype is brown eyes, but whose genotype is big B, little b. And if we want to say, what's the likelihood that they will have brown-eyed kids versus blue-eyed kids? Interesting. That's what we want to find out. Okay. First of all, when you do this and make space on your paper if you're taking paper notes, because this is going to be a list of things to do. I'm telling you the technique of this, and you will absolutely be tested on this. So make sure you're paying super good attention to this. Okay? Write down how to do this. Okay, first, draw a box. There it is. Then divide the box into four smaller boxes, like this. Okay? Now we're going to put one parent's genotype on top. I put the blue or the brown eye up here, so we'll put the brown-eyed parent here. It doesn't really matter. It works either way. Okay? It does not matter. I will not mark you wrong for doing it either way if I say one parent this, the other parent that. Okay, so put one parent's genotype on top like this with one letter for each column. Okay? Next, put the other parent's genotype on the left. So on the left side, like that. So one of its alleles for each row. Okay? So like this now. Okay. Now we're going to put an allele from each parent in each box. Okay, the capital letter comes first. It doesn't matter who gives you the capital letter. So um, looking at this box here, you get one big B from one parent, one little b from the other parent. So it's this, big B, little b. Okay? Again, the capital letter comes first no matter who it's from. It doesn't matter which side. It's just so that it's easier to see. We always put the big letter first so that you can see it's big B, little b instead of little b, big B. Okay, now let's see what goes here. So it gets a little b from this parent. So you kind of think of like this row. There's a little b goes in both of these. So a little b from this parent, a little b from that parent. So you get this. Okay, so down here, we're going this parent cross with this parent. So this is a gamete. This is a sex cell, really. It's an egg or a sperm. I guess this guy is a sperm. And it comes down here. And so we get big B, little b here. And what do you think we get here? That is right. It's little b, little b. Okay, so now your Punnett square is filled out. But what does it mean? Okay, so you're going to look at how many offspring have which genotype or phenotype. And remember, since you know what these genotypes mean, what phenotype they stand for, because we practiced that, you can now see how many of each offspring are which phenotype, genotype. Okay, so if this is 100%, this whole thing is 100% of the offspring, each one of these little boxes is going to be 25% of the offspring. So let's look at that. Okay, so looking at how many offspring have which genotype or phenotype. First, let's ask the question, how many brown-eyed children are there? Okay, well, first, you got to know that the brown-eyed children, they can have the genotype big B, or big B, little b. It can be either. They both look like brown eyes, remember, because big B is dominant. So even though there's little b here, the big B completely covers it up. Okay? So how many of these children, what percentage of these children are going to have brown eyes? Well, it's half of these. It's 50% because it's this one and this one. So 50%, 25% plus 25%, or you can look at the box as a fraction, 50% of them are going to have brown eyes. How many blue-eyed? Well, okay, so the blue-eyed genotype is little b, little b. So how many of those offspring have little b, little b? And you look, it's 50%, 50%. Because little b, little b, little b, little b. So you're gonna, on average, these people would have half blue-eyed children and half brown-eyed children. So that's the prediction that the Punnett square is making. Okay? Now, in any Punnett square, each square is 25%. So the how many of whoever question can only be this combination at this point in your biology career. You can actually make it do other things, but that comes later. So what I'll ask you, whenever I ask you how many, it's going to be one of these, 0%, 25%, 50%, 75%, or 100%. That's all there can be because it goes up in increments of 25%. Okay? Now let's check. These yellow peas are big Y, little y. Is big Y, little y a genotype or a phenotype? Well, it's the type of gene, so it's a genotype. The phenotype would be yellow peas. Okay. All right, so let's do an example of this. Yellow color is dominant over green in seeds, like in these pea plants. Okay, so here's yellow ones, here's green ones. Yellow is dominant. What letter would represent yellow alleles? Well, they always pick the dominant allele to be uh, to get the letter. So it's Y for yellow. So it's uppercase and dominant. What letter's representing the green? Now, before you say G, because that's the automatic thing, remember it's got to be the same letter as this. So it's going to be little y, little y. Okay? So it's lowercase. So even though it's green, it's little y because it's recessive to yellow. Yellow is dominant. Okay. So a plant, so here's our question that we're going to do. A plant that is homozygous for yellow seeds, so that means big Y, big Y. If I just told you homozygous for yellow seeds, you could figure out that it was big Y, because homozygous means two the same. Yellow seeds means uh, it's got at least one of these, at least one big Y. And since it's homozygous, it's got to be two big Ys. Okay, it is crossed with a plant that is homozygous, two the same, for green seeds. So that's little y, little y. What could their offspring look like? And plants have offspring. Come on, don't be judgy. Of course, plants have offspring. They've been having offspring longer than humans, for sure. Okay, how many of their offspring will be yellow? So here, I've set up the Punnett square already. So all you're doing is reading it off and remembering, okay? So how many of the offspring will be yellow? Think about what you know about the genotype and the phenotype here. Okay, it's 4 out of 4. So 100%, 100% of the offspring will be yellow because every single one of these offspring have a big Y in it. If it's got a big Y in it, it's going to be yellow because yellow is dominant. Even one big Y means yellow dominant. Doesn't matter if that Y, little y is there because it's recessive. The only way it would show up is if there's no dominant allele to cover it up. Okay, how many of the offspring will be green? Well, okay. Hopefully it's clear that if 100% are yellow, then 0% are going to be green. They're all going to be yellow. Okay, are these offspring homozygous or heterozygous? They're heterozygous. Hetero meaning different. So there's two different ones. So they're all big Y, little y. So they're all heterozygous. Okay, another example, just to make sure. Plants that are heterozygous, different for seed color are crossed. So what do their offspring look like? Okay, so there's big Y, and big Y, little y here. So this is one parent. This is the other parent. So they're both heterozygous. Let's see what their offspring look like. So as you make the Punnett square, big Y here, because Y from this parent, Y from this parent. For this one, big Y from this parent, little y from this parent. For this one here, big Y from this parent, little y from this parent. And from here, little y from this parent and this parent. Okay? So let's look at these guys. Think about what you know about the phenotypes of these, because look like means phenotype. Let's see. Okay, this is the same one, but scooted over so I could write over here. Okay, so what percent of the offspring are homozygous dominant? This is another kind of question I can ask you about these. So look and see how many of the offspring, remember the offspring are inside the box. How many are homozygous dominant? 1 out of 4, that's 25%, because each one of these little boxes is 25%. Okay, so 25% are big Y. How many are homozygous recessive? Homozygous, the same, recessive, little letter. That's this guy. So that's 1 out of 4. That's 25% also. Okay, and then what percent of the offspring are heterozygous? Heterozygous meaning different. 2 out of 4. So this one here and this one here, that's 50%. Okay, now thinking about phenotype, what percent of the offspring will be yellow? Now remember, yellow is dominant. So anything that has a big Y in it, even if it's mixed with a little y, is going to be yellow. So look at the offspring here. Think about what percent are going to be yellow. 3 out of 4, because this one and this one and this one have the dominant allele, and so they will be yellow. So 75% of the offspring will be yellow, and that leaves just this guy, green, because little y is the only time you're going to have green. Okay, so 1 out of 4, 25%. That's enough for today, and I will see you next time.